So this test, you have the beginning stuff, like how to grow microbes. I was like, I swore I had like 3 of these. Okay. How to grow microbes. We talked about like agar, the bacterial growth curve, oxygen requirements and all that. And then the second, and the majority of this test, we talk about how to kill microbes. Okay. So we start with the physical methods. They progressively get a little harder. Physical is the easiest, and chemical is a little harder, than antibiotics are even harder. So, like I said today, my main focus, I want to go over the physical methods. So, kind of just starting out. I remember I don't remember, but I know he told you guys to make a table, right? Okay, so what are the columns in that table? What are the different categories?
Method.
What?
Method.
Yeah, so I like to say the target. And what are some different options for the target?
Cell wall.
Yeah, so it can be the cell wall.
Do
you guys understand when I say
microbial target? Are those 2 different things? Because he gave that in class.
Did he? Oh, method means like heat, or that's the method. So heat is a method, alcohols are a method, different antibiotics are methods. Right now I'm just identifying these.
Okay.
But we'll get to that. You're fine. So the target, remember I'm trying to kill the microbe. So the target is what am I focusing my attention on to cause damage. So we can cause damage to the cell wall, like the peptidoglycan. Or I'm not going to give you like a full list because there's so many of them. Just like throwing out some ideas. What else do you think, you guys? Proteins. Okay, proteins are really important. Do you have any other ideas? Envelope, or we like to say cell membrane. So, because if we say cell membrane, we talk about bacteria and viruses, right? Because bacteria have a membrane. Then viruses don't technically have a membrane, but an envelope is the closest thing. So I'll just put membranes. There's one other really big one I can think of. Nucleic acid. Do you guys understand what that is? It's like DNA stands for deoxyribonucleic acid. RNA stands for ribonucleic acid. So it's just the genetic material of the cell. Okay, that's not a comprehensive list, but it's good enough to get us going, okay? Okay, what's another column in your table?
Cidal or static.
Cidal or static. What's the difference?
Cidal kills it, static suppresses.
So
Or not suppresses, but just keeps it from growing.
yeah, so cidal, if I have something that I consider to be, that's a shorthand version. It's actually called bactericidal. But if I have something that's bactericidal or cidal, it kills, okay? It's not going to give the microbe any chance to grow. But if I say something is static, that inhibits growth. I like to say it puts them on pause. So can they still replicate? Yeah, they can, but it's a lot slower.
Isn't this what they do for HIV?
Yeah, HIV, the reason why we wish it was cidal, but
Well, it just slows it down.
yeah, because that's just all we can do.
So
There are some drugs that we make them static on purpose, like antibiotics. Why would I make an antibiotic static?
Because you don't want to kill the good bacteria.
Yes, in a way, and then
Helps your body build up an immune to it.
yeah, because your body is what's gonna kill it in the end. So your immune system is what takes it out. So sometimes we'll have antibiotics that are cidal, sometimes static. And then all of our other methods, like our physical and chemical, that will apply too. And we'll talk about those. Okay, is that good? Okay, what's another column in your table?
Spectrum.
Yeah, broad or narrow, so spectrum. What does it mean if it's broad? Okay,
Bacteria or viruses.
this is like one of my favorite sections to tutor, but it's the hardest section to tutor because it changes every semester. A couple days ago, he gave me the chart of what you guys need to know this semester. So, broad versus narrow has a weird definition this semester. We're going to have physical methods, right? That's like heat, radiation, filtration, right? Then we're going to have chemical methods, like alcohols, halogens, metal compounds, phenolics. And then we're going to have antibiotics, like penicillin, chloramephenol, erythromycin, all of those. Now, if we're talking about physical or chemical, the definition of broad means what?
Isn't it covers like bacteria and viruses?
Yeah. So broad for the physical or chemical means bacteria and viruses. Okay. Is that good so far? What if I say broad for antibiotics? What do antibiotics affect?
Only bacteria.
They only affect bacteria. So how can I have a broad antibiotic if they only affect bacteria?
I guess it covers a lot of bacteria. And not
Yeah, what are the
just
two
one.
kinds of bacteria?
Positive.
Gram-positive and gram-negative. So broad antibiotic means it affects gram-positive and gram-negative. Is that okay? Okay, what if I said I had a narrow physical method or a narrow chemical method? What would that mean? Of what?
Like bacteria or
Bacteria or viruses, right? Because physical and chemical methods can affect bacteria and viruses. But if it's a narrow one, then we say for physical and chemical, it's bacteria or viruses. What if I said I had a narrow antibiotic?
One of the
One of the other of what?
positive.
Positive or negative? So gram-positive or gram-negative. So the definition changes a little bit based on what you're talking about. Does that kind of make sense? So just keep that in mind, especially when you go into class tomorrow. That when he says there's a broad antibiotic, that doesn't mean it affects bacteria and viruses. That means it affects positive and negative.
So like yesterday he gave the example of like alcohol.
Mm-hmm.
Like rubbing like hand sanitizer. That would be broad because it can kill bacteria too.
Yeah, that one is broad with an asterisk next to it.
Okay.
We might get into it, but alcohol, okay. I'm just going to put that on pause for a little bit, and I'll get back to it. Last column that we need to know. We have target, cytostatic, broad, narrow.
Antiseptic.
Yeah, antiseptic or disinfectant. And by the way, I can't spell for anything, so don't judge. I don't know if that's right. Okay, what is an antiseptic?
Something that lowers the
Yeah, so it can be put on living tissue. Give me an example of an antiseptic.
soap.
Yeah, soap, hand sanitizer, anything like that. Because I can put it on living tissue. Does it kill everything?
No.
No. Like, what does it say on the front of a hand sanitizer bottle?
99.9% of all germs.
Yeah, and I think that's really funny because one it says 99.9%, and then it says germs, and no one can define what a germ is. So that's their little tricky way of being able to put a really high number on there. So antiseptics, they don't kill everything, but they can be used on living tissue. Okay? What's a disinfectant?
Would bleach be one?
Yeah, bleach. So you never see me pouring bleach on myself, right?
Yeah.
And Lysol, anything like that. Anything I use on inanimate objects. So like this table, cleaning that glass. So I'm just going to write like tables, glass. I don't even know if that's how you spell it. Is it EL? Does that look right? No. Those are my hardest things, EL or LE. I don't know. Okay, so does disinfectant, does that kill everything?
No.
No. What is it called if it does kill everything?
Sterilize.
Sterilization. So if I sterilize something, I killed everything. Okay? There's no such thing as almost sterilizing something. You would just say, yeah, I disinfected it. Now there's another word that I just want to throw out there real fast. Another word that doesn't mean it killed everything. It's more of a method. A really famous microbiologist came up with it. His name's in it. Pasteurization. So Louis Pasteur created pasteurization. What's that? Do you know what it's used for? Foods like milk, yogurt, and all that. there's bacteria in there, right? But if I heat it up to boiling temperature, it's going to break protein bonds. And what's milk and yogurt made out of? Proteins. So if I heat it up too much, if you guys go home and boil milk, it doesn't taste the same. Okay, it's actually kind of gross. So what they do is they pasteurize it. Now, does pasteurization kill everything? No. So if I got a gallon of milk, didn't open the lid, and just kept it in my fridge, would it ever go bad? Yeah, because there are still microbes in there, and the refrigerator is doing a static effect. They can still grow, but it's a lot slower. Obviously, if I put my gallon of milk on the table, it'll get gross a lot faster than in the fridge. Is that okay? So pasteurization, it doesn't kill everything. It's just you're heating it up to almost boiling, so you can kill most things. In some countries,